So this is your introduction to Pfam, which is a super cool database that's one of the largest collection of families of proteins. Um, and it's going to be a critical piece for your final project. And again, this is very much specific to domains, and that's really kind of where we're focusing from the video we had before. Uniprot was more of a generic. Everything about it, Pfam, is way more directed towards domains, hence the family part. So let's just, to show you how to use this, let's just use one. Let's just do, this is one I did in an old class that I was a student. Let's do MH2 domain. Not that you care what this is or not, but we'll get to that. So let's just see what we want to see here. We're going to hit MH2 domain. Sometimes it does this with the new version of the site, so you have to just kind of do that. MH2 domain, it knew it. And so let's see what we get. Alrighty, so there we are. We see that there's 5,000 entries, but this is the one that we want. This is the Pfam session. So basically what these are, generically, they're typically found at the carboxy terminus of MAD proteins, or SMADs, which are conserved across species. They have to do with cellular development and whatnot. So there's a lot of organisms that have these, and you can see obviously this is where some of the information comes from. The MAD comes from this. It had to do with when it was found in Drosophila melanogaster fruit flies a long time ago. But we're not here to learn about that protein. We're here to learn about, or I should say this domain, we're here to learn about as much as we can about it. So this right here is the accession number. So if I ask you, for example, give you the MH2 domain and I say, what's the Pfam accession number? It's this one. Why? Because you can also see something that Pfam uses a lot of other different databases as well. So if we click on this, we see that we have a description which is really nice. This tells us kind of what this thing is, which is really, a great system here. We can look at something here called domain architectures, and this is really cool because this is the way that this particular domain has been organized and found in association with other domains. For example, there's 11,391 proteins that have the structure or architecture of an MH1 domain right at these particular loci, followed by an MH2 domain there. And there's 2,393 proteins that only have an MH2 domain and obviously don't have an MH1, and you get the idea. So all the way down the line, these are all the different structures, the way that MH2 has been found with other domains. And so that's helpful if that's kind of what you're looking for. So if I ask you how many domain architectures there are for MH2, the answer is 107. Let's go to taxonomy. This part's really cool. So a question you would be asked on your problem set would be, what sort of organisms is this domain found in, like which of the cells of which organism? And you can see this, it's only eukaryotes, and again the entire thing is green. That's not always the case for every protein, obviously, or every domain. But the beauty is you can kind of look around here and see which generic organism it's found in. You can search for it, or you can just click on it. So let's go, let's do Canis lupus. And so now you can see when I clicked on Canis lupus, now it's showing me that there are 78 sequences, and I want to see those. Let me go take a look and see those. So here is actually all of the MH2 domains from all the different genes only found in Canis lupus, and so that is very, very useful, and I can figure out whatever association I want or whatever I'm looking for or what particular gene. Obviously it makes sense that there's a lot of the SMAD ones. I can then, maybe this is the one that I want to look at and compare to something else. And now this is the actual domain right here, and the beauty is it kind of shows me, which is really nice, a lot more information. Here is the MH2 SMAD7. Here are other associations with homologous superfamilies and residues. I mean, there's a lot of different stuff here, which is really nice. So you can see here, I like the coloration. There's also gene ontology terms as well. Basically what that is that tells us what this thing does. and that it has been annotated to be related to things like regulation of templated transcription, protein binding, and transcription regulator complex. So that's what it does by the gene ontology associations. So let's just randomly click on this thing right here. And you can see here what this tells me is that it has a zinc binding site, a putative DNA binding site, which is nice as well. So I can get a lot of information from that. So now let me go back to here. I'll just go back to my overview from this particular spot here. As far as structures, let's go to structures for a second. So this is also cool because, again, it gives you the different structures of the association of this. As you can imagine, there's probably slight differences between the domain structure depending on what particular organism or cell it's in, and whether there's something that would be a mutant version, like for example this one. So that's really nice. And if you were to just click on a picture of it, you get a really beautiful, as soon as it loads up, 3D structure of it that you can again twist around and turn and zoom in and zoom out, go wherever you want. And again, like before, it has all of the same associations of domain, which is nice, and all the different chains. So this is a really nice piece here because it shows you all the chains of the domain itself. And so if we go back again to the overview, the curation is one other thing I want to show you about really quick. And this is basically the information related to the sequence ontology of this. And when I click on that, you'll see that this gives me more information, a lot more in fact, about this particular domain, which is really nice. And you can see here that it's probably a little small in your end, but I have a lot more information that I can look at from here. I can click on gene, excuse me, let that load up for a second. It's probably going to be a bit of a longer list. And you can see that these are all of, and we can get into candidate gene. If we do like a gene cassette, let's grab that one, or a mitochondrial gene. So we can get very, very, very specific with where this particular domain is represented in sequence ontology. And I know that sounded nerdy. So this is just an intro to Pfam, and again, it's related to domains. There's a lot more of this to know, except for a class like this, which is more intro, not necessarily. So you're going to be doing a bit of a web quest with this and answering questions about some domains. So on that note, good luck.